Welcome back to Bio 5. We're going to talk about DNA, RNA structure and function today. Learning outcomes are describe the structure of DNA molecule, explain the process of DNA replication, and list the major enzymes involved. Distinguish between structures of DNA and RNA, and state the roles of RNA inside a cell. DNA deoxyribonucleic acid is the genetic material. Within the genetic material, information is organized as genes. Genes are then clustered together in structures called chromosomes. There are three main functions. It replicates so that it can be transmitted to the next generation. It stores information, and also undergoes mutation and variability. DNA structure is a double helix composed of two strands that spiral around each other. Each strand is a series of nucleotides, and these nucleotides are composed of phosphates, sugar, and a base. DNA has a sugar deoxyribose and four different kinds of bases. Adenine represents A, thymine represents T, guanine represents G, cytosine represents C. There are complementary base pairing within DNA such that the A always pairs with T and G's always pairs with C. The phosphate backbone forms the upright double helix of DNA, and the base pairs comprising the rungs of the ladder-like shape. Adenine and guanine are the purines, the two rings, and the cytosine and thymine are pyrimidines, or the single ring. Here's a picture showing you the helical structure and the ladder-like step. The base pairs are A to T and G to C. A pairs with T with 2 hydrogen bonds, and G pairs with C with 3 hydrogen bonds. Over here, the picture showing you the pyridine and the purine base. The two strands of DNA are antiparallel. That means that they run in opposite directions, and this is due to the position of the carbon molecules on the deoxyribose sugars. So when you look at a double helix, one side will have a 5' carbon at one end, and then the other one will have a 3' carbon at the other. DNA replication. During the S phase of interphase during mitosis is when DNA is replicated. The double-stranded structure of DNA allows each original structure to serve as a template for the formation of a complementary new strand. DNA is thus coined as semiconservative, which means each new double helix has one original strand and one new strand. Over here is a picture showing you the mechanisms of replication. You can see that starts off with the parent strand, and now you split that, unzip it into two strands. The green one is a new one being grown. DNA replication is a relatively fast process. The first enzyme is called DNA helicase, which unwinds and unzips the DNA by breaking down the hydrogen bonds between the bases. DNA polymerase is able to add new nucleotides at a rate of about 50 per second, and rarely a replication error occurs. But if an error does occur, the cell has the ability to repair it via other enzymes. The leading strand follows a helicase enzyme, and the lagging strand forms short segments of DNA called the Okazaki fragments. Over here is a picture showing you that, the DNA polymerase, and then also the helicase unzipping the DNA. DNA like this seals the breaks in the phosphate backbone, and the two double helix molecules are identical to each other and to the original DNA molecule. A mutation is not always a bad thing. A mutation may be introduced variability by producing a new allele that alters the amino acid sequence and also the phenotype. Enzymes usually fix it, but sometimes it can't be fixed. The structure and function of RNA. RNA ribonucleic acid is made of nucleotides containing the sugar base. 4 nucleotides that make up the RNA molecule are the following bases: A for adenine, U for uracil, C for cytosine, and G for guanine. Most RNA is single-stranded. RNA is divided into coding and non-coding RNAs. The coding RNA is messenger RNA, mRNA, which is translated into protein. Over here is a comparison of similarities and differences between DNA and RNA. They're both nucleic acids, they're both composed of nucleotides, they both have sugar phosphate backbone, they both have 4 different types of bases, but differences are listed in the bottom chart. You can see the main difference is the bases. For DNA we have A, T, G, and C, but for RNA we have A, U, G, and C. DNA is double-stranded, whereas RNA is single-stranded. Over here is a picture showing you RNA by itself. There are 3 types of RNA: the messenger RNA, mRNA, ribosomal RNA, rRNA, transfer RNA, tRNA, and small RNA. Messenger RNA is produced in the nucleus. This type of RNA carries genetic information from DNA to the ribosomes and cytoplasm. Where protein synthesis occurs, and mRNA is a linear-based molecule. Ribosomal RNA, RNA, is formed from off a DNA template in the nucleolus. It joins with proteins imported from the cytoplasm to form a large and small subunits of the ribosome. During the process of protein synthesis, the ribosomal subunits combine to form a complex ribosome that acts as a workbench to generate proteins. Transfer RNA, tRNA, is produced in the nucleus, and a portion of DNA also serves as a template for its production. tRNA transfers amino acids to the ribosomes. At the ribosomes, amino acids are bonded together in the correct order to form a protein. There are 20 different types of amino acids used to make protein. Each type of tRNA carries only one type of amino acid, therefore at least 20 different tRNA molecules must be functioning in a cell to make a protein. Small RNAs are divided into several classes and are involved in splicing, modification, and regulation of mRNA. Small nuclear RNAs, snRNAs, and also the small nucleolar RNAs, snRNAs. We also have microRNAs, miRNAs, which are attached to the mRNAs in the cytoplasm, and they prevent unnecessary translation. We also have the small interfering RNAs, siRNAs, that can bind to mRNAs, and they can then prepare mRNA for degradation and breakdown.